I was planning on it.
Get ready to
One little dungeon dragon.
talk some more about iron and macrophages. Are there any questions from the first hour? No? Okay. All right. So I think where we left was macrophage in the spleen was taking up an aged red blood cell, engulfing it, digesting away the hemoglobin to release heme. Then heme oxidase releases the ferrous iron. And then the excess ferrous iron is going to be effluxed out of the macrophage, oxidized by ceruloplasmin, and then it can bind the transferrin molecule. The transferrin then can take this iron back to the bone marrow to support more erythropoiesis. Right? Now, that's the case for normal red blood cell turnover. What happened to Isaac's red blood cells? Where were they being lysed? So some of them were probably, well, a lot of them were probably being taken up by this pathway, right? But there's one key laboratory value that you may have overlooked, but that was the haptoglobin. So there are two molecules, haptoglobin and hemopexin. And these are molecules that can scavenge iron as a result of intravascular hemolysis. That is, the red blood cell blowing up somewhere besides within a macrophage endocytic vesicle. Okay, so the haptoglobin binds free hemoglobin that's released from that intravascular hemolysis. And when the haptoglobin binds to hemoglobin, then that can be taken up by macrophages through a receptor-mediated endocytosis process. Okay. Likewise, the hemopexin, that can bind up free heme that would result from intravascular hemolysis, and similarly recover that heme iron through receptor-mediated hemocytosis, receptor-mediated endocytosis of the hemopexin receptor complex. Okay. And then once that heme is in the macrophage, then there's a heme oxidase which will release the ferrous iron. Okay. Let's see, just a couple things that I want to mention before we leave the macrophage compartment here. And that is that in addition to this getting iron from a red blood cell, the macrophages also have transferrin receptor, and if they're not in the active business of eating a red blood cell, they can get iron through that same mechanism of transferrin receptor-mediated endocytosis that all other cells can. I also want to mention hemosiderin. So hemosiderin, this is that aggregate form of ferritin, and I think of this as being most important in the liver because the liver is that, it's the storage sink of iron within our body. Normally, it wouldn't be in a macrophage in abundance because the macrophage is going to be exporting iron out to support erythropoiesis. However, there are conditions where if you have abundant hepcidin not only is blocking efflux of iron from the gut epithelial cells, but it's also blocking iron export from macrophages. So in a condition where you have elevated hepcidin, there you're going to still have red blood cell turnover, but the iron is going to be stuck within the macrophage, and there you could get the buildup of hemosiderin. Okay. Okay, I think I mentioned this. Why don't we talk then, since we just were discussing hepcidin, segue into a discussion of iron in a hepatocyte, in a liver cell. And with a hepatocyte, you kind of have to think of two things. First off, the hepatocyte is going to need some iron So it will take iron up out of circulation through transferrin binding to the transferrin receptor and receptor-mediated endocytosis, and then the steep reduction of the iron and transport out of the endocytic vesicle through the DMT1, the basic same story of other cells. Okay, there are a few peculiarities of the iron in hepatocytes, and I guess one, I'm not sure why it wasn't shown here, but there's an equilibrium between ferritin and hemosiderin, that kind of bigger aggregate of the ferritin molecules. Hepatocytes are the organ that excess heme is excreted through, right? So when heme is taken up by the hepatocyte, or the heme will be converted, of course, into the monoglucuronate and then the diglucuronate, and then it's exported through the bile. So I guess that's after it's been exposed, or after the iron has been released, so maybe not so relevant to today's topic. Okay, so I'm going to switch and discuss kind of a second function of iron in the hepatocytes, and that is the hepatocyte's role in synthesizing hepcidin and producing this endocrine controller of iron homeostasis. So the production of hepcidin by the liver is largely dependent on transferrin interacting with the transferrin receptor, but it doesn't involve the receptor-mediated endocytosis and the release of the transferrin. It's more of a signal transduction cascade that is initiated by the transferrin binding to the transferrin receptor. Okay, so just to review, the hepcidin molecule, when that protein is produced, it goes into the circulation and then it binds to ferroportin either on a duodenal gut epithelial cell or on a macrophage, and it causes the ferroportin to be internalized and ultimately degraded, and that causes decrease in uptake of dietary iron, as well as sequestration of iron within the macrophage compartment. Okay, so let's look at where that hepcidin comes from. Hepcidin is synthesized in the liver, and it's encoded by the HAMP gene, and the HAMP gene promoter has multiple different kind of signaling cascades that are entering into it. And really it's going to be sensitive to three kind of important things. One, the one that we'll spend the most time with is iron status. So there's a transferrin receptor signaling axis that enters into the HAMP promoter. Inflammation is also a key driver or a key regulator of the HAMP gene expression. And then the erythropoiesis, or how much red blood cell synthesis is going on, is also key. Okay, so I'm going to talk I don't know why I've got. Okay, I guess I think I'm going to talk more about hepcidin in the context of the pathologies. So I apologize for that. I got a little mixed up here. I guess it's all right here on this. Sorry, I got a little mixed up. Okay, so the HAMP gene, the promoter, is sensitive to these three kind of different signaling axes. And the role of the HAMP gene expression is to promote the sequestration of iron, or to reduce the amount of circulating iron around by preventing macrophage efflux and gut epithelial cell efflux. Okay, so the transferrin receptor, in addition to having that the receptor-mediated endocytosis, it's also linked to some signaling cascades through the HFE gene, and there's a big complex of other proteins that will signal to the HAMP promoter and cause an increase in expression of hepcidin and a blocking of the uptake of iron from the diet. So basically the transferrin receptor in the liver, it's promoting iron homeostasis. When you've got a lot of transferrin binding to transferrin receptor, hepcidin expression increases, and that then prevents you from taking up more iron. So that's kind of a way to maintain iron homeostasis. Independently of iron status, the interleukin-6 receptor, that can cause hepcidin expression related to inflammation. And inflammation can be due to infection or due to some other, disease status. And the idea here is if you're septic, if you have bacteria in your blood, you don't want to give those bacteria iron because bacteria require iron for their metabolism. So by coupling the hepcidin expression to inflammation, that will then allow an infected person to not give the bacteria the iron that they need to survive. Okay, and then erythropoiesis there, when you're doing more erythropoiesis, you're going to be needing more iron. So this is a negative influence into the hepcidin gene expression. So the more erythropoiesis is going on, the lower the hepcidin production will be because anticipating a greater need for iron. So we'll revisit some of this when we look at some disorders of red blood cell homeostasis and iron metabolism. Okay, so let's temporarily leave iron in the hepatocytes and discuss briefly iron in the kidney. So the kidney, this is our main controller or one of the main controllers of erythropoiesis through the hypoxia-inducible factor regulation of the erythropoietin cytokine. So erythropoietin acts at multiple points on the erythroid lineage to promote proliferation and differentiation into red blood cells. And the erythropoietin expression, this is dependent on hypoxia-inducible factor. Hypoxia-inducible factor, so we don't have to go through this whole pathway of the prolyl hydroxylation and everything. I'll leave it for your review because I think we've seen it before. But prolyl hydroxylase, so this is, we think it just sticks an OH on a proline and that's it. But it really it's dependent on alpha-ketoglutarate, and it's dependent on molecular oxygen, and also dependent on the availability of iron. So it's kind of got this. In addition to an oxygen sensor, it can also function as an iron sensor and as a TCA cycle intermediate sensor. Okay, so this, again, is one of these big summaries that I could probably just do the whole 2 hours of lecture on one slide if I wanted to, but I don't think that would be good for anybody. So I think I'll leave this slide for you to review, and if you have any questions about it, come and visit with me, and I'll be happy to discuss. Okay, so we've talked a lot about iron as a necessary component of hemoglobin and a little bit about myoglobin. Just to round this out, there are many other uses for iron within cells. So heme iron is used in complex 4 of the electron transport chain. There are also iron sulfur complexes that are used in complex 1 and elsewhere. So just sustaining respiration in the mitochondria is a key function of iron. There are also a lot of DNA repair enzymes, ribonucleotide reductase, that enzyme that converts a nucleotide into a deoxynucleotide. That's an iron-dependent enzyme. Primase, that's an iron-dependent enzyme. So quite a lot of other things are needing iron for diverse purposes in all cells of our body, but the amount is minute compared to the amount of iron that is dedicated to synthesizing heme. Okay, iron deficiency. So we'll talk first about some different conditions of iron deficiency. Gesundheit. And then we'll talk about some conditions of iron excess, and then we'll conclude with a case and some clicker slides. Okay, so iron deficiency causes anemia because your ferrochelatase doesn't have a substrate, so you can't synthesize heme, so you can't synthesize hemoglobin, so you can't synthesize red blood cells. Okay, so there's some common symptoms for all anemia, like fatigue and shortness of breath and pallor, etc. Iron deficiency anemia has some specific features. The anemia will be microcytic and hypochromic, right? So just reflecting the lack of hemoglobin. There are some specific symptoms that are associated with iron deficiency anemia that can distinguish it from other kinds of anemia, and mechanistically, I don't really have a lot to add to that. So if we're drawing that on PBL, that'd be one of those arrow, question mark arrows. But I'll leave that to your own curiosity and investigation. Okay, so the iron deficiency, how we can get iron deficiency, we can get it either by not eating enough iron or by needing too much iron, I guess. So just by blood loss, either by physiological blood loss or pathological blood loss, that can cause an iron deficiency because you're going to have a huge increase in erythropoiesis to compensate for the loss of the red blood cells. Okay, so there you can, be eating an adequate amount of iron for normal circumstances, but you have this transient need for greatly increased erythropoiesis, so that can cause a transient iron deficiency. Like if you're in a hugely anabolic state as a child or in pregnancy, that can again have a heightened need for iron and can cause a transient iron deficiency. You can also get iron deficiency through inadequate supply, so either not consuming enough iron in the diet, or alternatively, if you have some problem with the duodenal epithelial cells, that can manifest as an iron deficiency because the duodenum is the site of iron uptake. Okay. All right, so let's consider where the iron goes in some of these conditions of iron deficiency. And we'll start out with the normal stores. So these different boxes here, these are reflecting iron stores in erythroid cells, in red blood cells, in the liver, and then the dark blue, this is the macrophage iron. Okay, and then there are some different lab values here, and I think that some of these Dr. Saxena probably discussed. Let's see, transferrin saturation. This is in super tiny type here, so it's hard for me to see, but transferrin saturation, I think, is right there, right? And that's about 35% up to 50% or down to 20%. Then mean corpuscular volume is here, so keep an eye on that one. This RBC ZNPP, this is red blood cell zinc protoporphyrin. And this, I should have mentioned this maybe on the heme synthesis lecture, but at some low level, the ferrochelatase, instead of putting an iron atom in the middle of a heme ring, it puts a zinc atom in between there. So that RBC ZNPP, that's like heme except with a zinc instead of an iron. Okay. Then we have hepcidin levels up here. And yeah, I think so. Those are the main ones, I guess, that I'd like you to keep track of. Okay, so let's take this healthy person with a typical diet that is replete with iron and remove the iron from the person and see what happens. So as you can imagine, you lose first the iron stores, then you start to lose erythroid iron, and then ultimately you develop a condition referred to as iron deficiency anemia. Okay, so as you're losing the iron, the first things, or one of the first things that happens, is you start to get a decrease in hepcidin, right? And this is just exactly what you would want to happen, because you want your duodenal epithelial cells to take up as much dietary iron as possible to reduce or to ameliorate the deficiency. And going right along with that, the transferrin saturation, which, okay, down here, that's also decreasing right along with it. So first you're losing your stored iron, your hepatocyte and macrophage stored iron, and then you start to lose iron in the erythroid compartment. And then when you start to really lose red blood cells and become frankly anemic, it's not until that point that you see a change in the mean corpuscular volume. So that's kind of a late feature of the microcytic anemia that's associated with iron deficiency anemia. Let's see, that zinc-containing heme, the RBC zinc protoporphyrin, as your iron decreases, the zinc-containing heme, that goes up as well. So that's another biochemical indicator of low iron stores. Okay, is this all make sense? Okay. Now, if you looked at the bone marrow, or if you looked at the blood and the bone marrow of a patient with iron deficiency anemia, you would see small cells and pale cells. And again, this dark ring around the red blood cell, I've heard from the pathologist, that's supposed to be about 1/3 of the diameter of the red blood cell. And you can see, here it's much less than that. So this is a patient, some of the hematological parameters are shown here. So this is a pretty severe anemia, severe iron deficiency anemia. If you look at the bone marrow and do a Prussian blue stain, this is a Prussian blue stain in normal individuals, and the magnification is a little inconsistent here, but normally in erythroid precursors in the bone marrow, you should see some blue dots reflecting iron in those precursors that is going to be dedicated towards heme synthesis and hemoglobin synthesis. And in the erythroid precursors from our patients' bone marrow, you see that's absent. Okay, questions on iron deficiency anemia? So how would you treat this patient? Iron supplementation, probably? Okay. Yeah, that's right. Yeah, iron supplementation is cheap, and if that's the problem, she'll get better from it pretty quickly. Now, what if you have a patient with an apparent iron deficiency anemia and you replace iron and the patient doesn't get better? Now, well, you might look at the duodenum and see if there's an absorption problem, right? And if there is, then you might want to give intravenous iron. And if that doesn't solve the problem, then you might have a condition called iron refractory iron deficient anemia. And iron refractory iron deficiency anemia presents as, I guess, a more severe microcytic anemia with more anemic and more microcytic. And most of the hematological values here are pretty similar to iron deficiency anemia, except for the hepcidin. Now, in a normal person, in a healthy individual who had an anemia this significant and had transferrin saturation that was that low, what would the hepcidin values be? Well, let's look. They should normally, in iron deficiency anemia, they should be very low, right? Because your transferrin saturation is low, so you get lack of interaction with the liver cells, transferrin receptor, and lack of production of the hepcidin protein. Right? So why in this condition is the hepcidin high when it should normally be low in order to restore iron homeostasis? Any thoughts?
Because there is iron, but it's just happening later in the way.
It's not. There's no iron. And you can see like transferrin amount is very low, and yeah. So iron is legitimately low. And the reason is because there is a mutation in the one of the receptor complexes that signals into the hepcidin gene that leads to an overactivation or a constitutive activation of the gene. So you've got the transferrin receptor that binds to iron-bound transferrin here. Normally that sends a signal through SMAD proteins, and SMADs are kind of similar to JAK-STAT coupled receptors. Anyway, there's crosstalk between the transferrin receptor and the bone morphogenic protein receptor that leads to a SMAD signal to the hepcidin promoter. Okay, so kind of a lot of alphabet soup there. There's a protein whose common name is matripase. It's also got the TMPR SS6. So that's one of the proteins that makes up this bone morphogenic protein receptor complex. That can, there are gain of function mutations in it that will cause this signaling to be constitutively active and to be decoupled from any transferrin receptor. Okay, so even in the absence of transferrin receptor signaling, you get that SMAD signaling into the HAMP gene promoter and excessive production of hepcidin when you have very low iron levels. Okay, is that clear? So this is a gain of function mutation that causes overactive signaling of the and inappropriately high expression of hepcidin, and that leads to the more severe anemia because you've got this presumably since birth, because it's an inborn, it's a mutation that you carry for your whole life. Okay, so questions on iron refractory, iron deficiency anemia? Yes?
Would IV iron still treat this?
I think it could, but it's going to be sequestered in macrophages. The IV iron, that could bypass the inhibition of hepcidin in the gut cells, but it's not going to bypass the sequestration of iron in the macrophages. So yeah, I think it might have some role, but it's maybe a little more complicated than that. Okay, so let's look at one more form of anemia has an iron deficiency component to it, and this is anemia of chronic disease. With anemia of chronic disease, if you look at some of the parameters down here, it's not as severe as the iron deficiency anemia, and it's marked by a specific deficiency in erythroid iron without a loss of iron stored in macrophages and iron stored in hepatocytes. So the problem with anemia of chronic disease is similarly to iron refractory anemia, where you get an inappropriately high level of hepcidin production, and that's going to prevent uptake of dietary iron as well as sequester iron within macrophages. The difference is that, the anemia of chronic disease, that is, well, it's called chronic disease, right? But you don't have it from birth as you would the mutations in matripase from an iron refractory iron deficiency anemia, right? So this would be just, you get cancer, you start getting anemia, you block up the iron where it is, but it's a little bit different than having a elevated hepcidin from your entire life. Okay, so that anemia of chronic disease, as you may guess, that is due to one of those other inputs into the hepcidin gene promoter, the interleukin 6 receptor. So the interleukin 6, this is a cytokine receptor that is coupled to JAK-STAT signaling. And in conditions of cancer or sepsis or other like chronic inflammation, you get heightened signaling of IL-6, and that leads to activation of the hepcidin gene and increased production of hepcidin. And if that's sustained, that can lead to that anemia of chronic disease. Let's see. So I think that's, and the rationale there is, I think I mentioned this once before, but in the case of a infection, say, these bacteria, their proliferation is dependent on iron. And the idea behind the increased hepcidin production in the presence of chronic inflammation is that you're going to lock down all the iron within the body and prevent any iron from being available to the microorganisms, and then that can allow the antibacterial defenses to clear them from the body. Okay, is that all good? So going back here, would you be comfortable distinguishing anemia of chronic disease, iron refractory, iron deficiency anemia, and iron deficiency anemia? Okay, the clinical presentation and then the lab values, I think they're pretty straightforward, but spend a little bit of time on it. Okay, so I'm going to switch now and talk about conditions of iron excess, and I'll use the same table. So there are several different conditions of iron excess shown here. I think, let's see, this one is transfusional overload, and we've talked about what we've mentioned it, I think, as a potential complication of multiple transfusions. As far as our session here, let's focus on the hemochromatosis that's caused by mutations in the HFE gene. There are a few other hemochromatosis, but let's focus on this one. Okay, so the hemochromatosis is an inborn error of metabolism that's associated with abnormally low hepcidin production, even when you have abundant iron. And in terms of the course of the disease, it has a long, like a non-symptomatic form, or a long period where the only way that you could tell it was there would be to sequence the gene, right, and identify it through a gene mutation. Okay. Over time, you get some biochemical presentation of the disease or some abnormal laboratory values that you could identify that would precede the symptoms developing. And the lab values you could look at are the transferrin saturation, which would be abnormally high, and then serum ferritin would be, that would start to go up as iron accumulates over the years. And then ultimately, you're going to get some symptoms of kind of constitutional symptoms of fatigue and hedonism and what have you. And then some specific symptoms related to specific organ damage of the liver and kidneys and hearts from oxidative damage due to Fenton reaction, essentially making heightened oxidative stress on the tissues. Okay, so the most common cause of hemochromatosis is through mutations in this HFE gene, and the HFE protein makes a complex with the transferrin receptors that's necessary for propagating that signal to the bone morphogenic protein receptor and then ultimately to the HAMP gene promoter. So if you have those HFE gene mutations, you then don't get hepcidin production even in conditions where you have high transferrin saturation or high iron. Okay, so if you look, let's see, I guess we had back here. So if we have abundant iron normally, or like, let's say in the case of the transfusional overload, we have normal, we have high iron as a result of the transfusion. The normal response to that is going to be to increase hepcidin, right? In hemochromatosis, we've got excess iron. You can see that lot of liver iron excess, but we don't have that hepcidin response. Okay. This doesn't generally result in like a polycythemia, although you can have a kind of high normal or just on the edge of abnormally high hematocrits in this case. Okay, yeah. So the problem, the most common form of hemochromatosis is by mutations in the HFE gene. You can also hemojuvelin, which is one of that's that HJV in the bone morphogenic protein receptor, is another common form of hemochromatosis, and there are other known causes of hemochromatosis in different members of this signaling pathway. Okay, so with chronic hemochromatosis, normally when you have abundant iron and a high transferrin saturation, that should result in production of hepcidin that's going to block the uptake of more iron. But in the cases of HFE hemochromatosis, you're decoupling that hepcidin production with the transferrin receptor. Okay, so you no longer get hepcidin production when the transferrin receptor is tickled, so you get basically unregulated uptake of dietary iron, and that can accumulate then and cause organ damage. Okay, now it's, I guess, important to note that the symptoms of that hemochromatosis, like, fatigue or just kind of non-specific feeling like crap, shortness of breath can be one symptom of hemochromatosis. That kind of recapitulates some of the symptoms of an anemia, right? So, be careful and look at the blood, not just the symptoms. Okay, questions? Okay, should we look at a look at a case? I think I gave you this first page of the case, right? Any impressions, or need some more time? Well, what would you do with the flight surgeon? You try a clicker slide? Okay, so this is probably fairly straightforward, immediately after a lecture on the topic, but okay, so why wouldn't it be iron deficiency anemia, or what would be arguing against iron deficiency anemia? Yeah, the serum ferritin being high. I would say also on the previous page, he had a normal or high normal hematocrits. And then the symptoms that he had were, it was some of the same symptoms of an anemia, but he also had some kind of more severe stuff going on that was affecting more organ systems. Okay. So he had hemochromatosis. Okay, yep. Best answer is hepcidin in circulation. Yep, so ferroportin both on the gut cells and on the macrophages, that's going to be increased because of the lack of hepcidin. Heme bound to hemoglobin, I mean, this is, maybe arguably this would be elevated because he had the patient had the high normal hematocrits, right? But I think, yeah, most significant one is going to be the hepcidin in circulation is going to be way down. Okay, so last question here. Okay, now iron restricted diet, I think that might be reasonable. However, like he's got problems now, and if you do a diet change now, that iron's still in his body for a long time, right? So therapeutic phlebotomy would be the best choice for for a patient who's suffering some pretty significant symptoms right away. Yep.
So you would do therapeutic phlebotomy acutely, but would you continue to do that over time for the patient, or would like a chronic treatment be to restrict the iron in his diet?
The phlebotomy is, I think that's done every few months in situations like this. Yeah, iron is so abundant in a lot of foods, and generally we're eating way more than we're going to take up. So I think an iron restricted diet is kind of tough to maintain. Yeah.
Since he's living at altitude, would that cause any problems with that in this case, or no?
That, yeah. I don't know. What do you think?
I would guess so.
Um,
Not a doctor.
he's going to have less oxygen around, so less availability or less potential to make oxidative stress, right? If you're going to have less oxygen. If he's kind of chronically hypoxic. If he's a flight surgeon and goes in airplanes a lot, he might be in like a dynamic environment and have some, he might be like breathing oxygen sometimes. So I don't know how to answer that. Sorry, but it was a lot of words to get there, right? So, yes.
Is the increased ferritin the because when you have so much iron, do your cells just kind of like start pooping out the stored form?
Yes, yes. And it's going to get worse as you get more damage to tissues. You know, you're going to have cells dying and they're just dumping out their ferritin around. But yeah, before they do that, they're kind of leaking it out. Yes.
That was kind of my question. So on the last one, the clicker question was the increased hemosiderin. So wouldn't you also have hemosiderin too? Just wouldn't be as.
Well, this question is, I think, what would be decreased?
Oh, okay.
Yeah, so hemosiderin in hepatocytes would likely be markedly increased in this patient. Good question. Any others? Okay, so this patient had HFE hemochromatosis, and he had therapeutic phlebotomy, and symptoms resolved, and he returned to treatment. And this is kind of a cool journal. It's a lot of like high altitude medicine and space medicine. So, okay, here's again, you know, this is about the third one of these nice comprehensive summaries. So just pick whichever one you like. Okay, are there questions? Okay, I know many of you have PBL, so I'm okay with leaving a few minutes early here. Okay, bye. I have to go to PBL right away, so give me like until end of the day to get the clicker slides up, if you would. If I have a few moments, I'll get them up before that, but don't look for them immediately.
You should do like a scream at him. Make it so loud, everyone. Oh, sorry. Good luck, you guys. Treatment plan. I didn't add anything about like what type of foods. To make me feel better, I'll just say, hey, other groups do the same thing, and she'll be like, oh, that doesn't make sense.
Glad you didn't lose your wallet. I would help you find it, but I feel like you did so.
To do my face presentation.
You're a rocket.
I didn't say
Still like last night.
education.
I watched that.
Asian food to avoid.
And then I'll lock in.
I didn't want to like be repetitive. I'm telling you,
Like, okay, fine.
they should
That
know
actually
it.
is, but I'll do it again. People, I mean, it's all, I feel like repetitive. Just avoid triggers.
Avoid triggers.
Yeah. See you later. It's just how we luck out. I feel like it's really nice. No, I was like, okay, that works. It's fine. It's fine. It will be fine if they avoid those things.
I'm actually reading some can exercise induce it.
Y'all go you guys feel it.
PD in your muscles are different other tissues, and it's weird. So like,
It's like a different
oxidative stress.
isoform.
Yeah, it's weird. It's like you can get lactic acid from that.
Yeah.
But like from your muscles, you don't function.
Yeah.
Say isoform increases it. So if you did have it, you're like, yeah, you can go for 10-mile runs. Fine.
Exercise would probably makes everything better. If I've learned one thing about medicine.
All right. I had to take a. Let you know in case you want.
I don't know, Hank, I understood it until you started asking questions.
So it's decreased.
No, that's okay.
Decreased his iron diet.
Oh, yeah, decrease.
On the last question with the iron, it was mentioned like anemic symptoms. Are you getting anemia with these iron excess?
No,
Just the fatigue is from something else?
it's from oxidative stress to organs and yeah, causing damage. So there might be
Oh, the
damage
Fenton
to
reaction you said.
Yeah,
Can be
yes,
the
exactly.
thing there playing
So
in.
that's-
Okay.
That could be damaging the lungs, and he's got plenty of red blood cells and hemoglobin, but just not able to take a breath or the muscles are crapping out.
Okay. So the increased iron in his red blood cells then, that could be causing them then to,
this Gunner asking?
be more likely to be like methemoglobin or something potentially. So that decreased oxygen carrying capacity and anemia through that or no?
Now, if you'll notice just like the size of the red bar here,
Yeah.
this is the erythroid iron, right?
Okay, yeah.
So that goes down in an iron deficiency.
Botros
usually have some. Why do not?
Right, so you get like, you know, maybe.
Isn't hemochromatosis this one that goes down?
water.
Am I looking?
No, that's
Oh, is that why it's
Oh, okay.
Yeah. Yeah, so the iron overload doesn't.
Make you have more in your red blood cells.
Yeah, that
necessarily.
Can you look at my
I was feeling a little dizzy.
Like the pipe surgeon here, he had a high normal hematocrit, so, you know, on the edge of
able to test that right here?
high, but not like abnormal.
Yeah, okay, I see. Alright, that's good, thanks.
Y'all
So on slide 5, it says RE cells, is that right?
Good or bad?
reticular endothelial cells.
Endothelial.
So that's the macrophages of the spleen.
Gotcha, okay,
Yeah.
that's good. That's what I was hoping it meant. And then another question, so with the macrophages as well, I guess we can go back to that same diagram. So it shows like ferritin so in these cells and also in the liver, and an arrow out to serum ferritin.
Yep.
Is there then mechanisms to bring that into other cells, or do they have to kind of dump their iron out before they go back in? I'm just imagining these balls of iron just flying around.
Yeah.
What do they, I don't know what they do.
That, you know, I honestly don't know the fate of the ferritin after it goes out of the cell. I imagine there's gotta be some kind of a way that it gets retaken up, but I have not seen how that happens. And yeah, that's a good question though.
Okay.
Yeah, okay, I'm gonna do some research on it.
Yeah, okay, thanks.
You're welcome. Hi.
So he had high hemoglobin and high hematocrit. Does that just due to the hypoxia that he's experiencing going to the new place?
Yeah, I mean, that could be from the hypoxia. It could be because iron is in no way limiting his erythropoiesis. So he's like, you know, he's at maximum erythropoiesis as far as the availability of iron. And that is something that is limiting to erythropoiesis. But he's not like polycythemia vera levels of high hematocrits. It's kind of, there is a homeostasis, it's just nudged a little bit higher. Okay, yeah, perfect.
Sounds good, thank you.
Yes.
You also, I think, answered my other question about the thalassemia thing and how it would, I think it's the, well, then I Googled it, there's a thing called, what was it called? Erythroferon.
Okay.
That's something that's made in the erythroblasts. So they're basically saying that then the high erythropoiesis turnover rate in like a thalassemia is causing the increase in this erythroferon because there's so much erythropoiesis going on, and that goes and inhibits in the liver the hepcidin. Yeah.
Okay, so I think that's